Prior to joining our faculty in 2004, Mary held faculty appointments at the Institute of Psychiatry and King's College in London. As many of you know, Mary's outstanding program of research aims to understand the pathophysiological basis of bipolar and other mood disorders. She uses neuroimaging techniques to discover alterations in brain circuits, emotion regulation, and reward processing that are associated with these disorders. Her research also focuses on identifying disturbances in the neurodevelopmental trajectories of these brain circuits and the extent to which these neuroimaging techniques can be used to identify biomarkers for disease risk. Her latest work examines how neuromodulation techniques can be targeted on such biomarkers to develop new interventions for individuals with mood disorders. Mary has served as the principal investigator or co-principal investigator for more than 15 federally sponsored research awards. Her impressive publication portfolio includes over 220 peer-reviewed articles, many of which have been highly cited in the field, and a number of influential reviews. She has, of course, been recognized for her scientific contributions and accomplishments through the receipt of multiple awards and honors. I'll just name a few. She's received the prestigious Joel Elke's Research Award of the American College of Neuropsychopharmacology and the Brain and Behavior Research Foundation Colvin Prize for Outstanding Achievement in Mood Disorders. She's an elected member of the American Society of Clinical Investigation, and she's served as president of the Society for Biological Psychiatry. As all of you know, she's also an extremely talented and gifted and generous teacher and mentor, and she has served as a mentor, and her laboratory has served as the incubator for the careers of a number of promising trainees and early career scientists in our community. So although applause is at a distance today, except for the few of us who are able to be in this room, please join me in welcoming my esteemed colleague, Dr. Mary Phillips, as our keynote speaker for this research matinee. Mary. I failed to say that you should, we'll have question and answers throughout the day. You can submit your questions at any time by texting PitSci to 37607 to join the poll. You'll receive this message. And let me just leave it up here for a second so everybody can write it down. And you can actually go ahead and text that now, and then at any time during Mary's talk or during the subsequent sessions, submit a question.
Well, thank you very much, Dr. Lewis, for that very kind introduction. And I just want to say it's my honor and pleasure to be able to give this presentation today. And I'm really going to go through a journey of the work that I've been doing in the lab, and also a little bit of background about me. My first task of today is to actually get my slides on the screen, and I'm going to try and do that now. So here we go. So the title of my talk is Inspiration, Loss, Persuasion and Persistence, How I Started and Where I'm Going in Mood Disorders Research. And I really want to highlight that this is really sort of a series of points which I think are some of the most important in my research career, and how sort of pivot points during my time in this area have made me change my mind, have had sort of changed my decision pathways, and have also highlighted new directions in my research. As most of you know, my research really focuses on understanding neural circuitry, neural networks, underlying predisposition to risk for and development of mood disorders. And more recently, I've been interested in using that knowledge, the discoveries we've made in the lab, to try and develop better treatments for people suffering from these disorders. That's really what I'm going to say at this point about the overarching point and area of my research. But really what I want to do now is go back on a journey for me personally as to how I got into all of this, where my inspiration points were, who inspired me, and what made me change my mind, what made me change directions as I went through these pathways. So I have to say it probably all started when I was about 4 or 5. I became totally inspired by a wonderful person, Elizabeth I, one of the Tudor monarchs in the United Kingdom, Great Britain, England as it was known then, and probably it's going to be known again. And Elizabeth I was an inspiration to me, not only because she was one of the most powerful, became one of the most powerful female monarchs, but because of the opposite. It's because she came from such a relatively humble, but then quite a disastrous background. So let me just tell you a little bit about her background. And remember that I was 4 or 5 years old at the time when I started to learn about this. So I was totally impressed. So what I'm showing you now is a picture of Princess Elizabeth. I think she was about 13 or so at the time. So she looks very regal and very grand, but I suspect that many of you will know that she was actually the product of Henry VIII there at the bottom, and Anne Boleyn, Henry's second wife. Now, in those days, people didn't believe that women could be leaders. So Henry was very disappointed that Elizabeth, his child with Anne Boleyn, his favourite wife at that point, was a girl. And after a couple of years, when Anne was only in her 30s and Elizabeth was only 2.5 years old, Anne was executed. She was beheaded on charges of treason, which have since been questioned. Then Elizabeth was declared illegitimate twice. She was declared illegitimate and then she was reinstated, and then her younger brother, who was then King Edward VI, made her illegitimate again. Moving forward, Mary, I'm showing you there, he looks rather stern. That's Mary I, Mary Tudor. She was only the second female monarch in the UK's history. Now, at that point, we're talking 1553, 1554, England was in a terrible state. It was almost on the verge of civil war. Catholics and Protestants were against each other. Mary inherited the throne. She was a staunch Catholic. She was very worried about keeping on to her throne, and she was the subject of many rebellions. The most famous of all was the White Rebellion, which caused her so much grief and anxiety that she had her younger sister Elizabeth placed in the Tower of London, ready to be tried and executed for treason, despite the fact that Elizabeth probably was not guilty and was part of the plot. Elizabeth managed to escape being beheaded. You can imagine how terrified she was. She was about 21 years old in the Tower of London. She knew many of her friends and colleagues had already been beheaded. She was there alone, but she managed to escape on the basis of 2 things: her intelligence and wit, and also because at the time it was thought to be probably not a great idea to execute potentially a future Queen of England. So she survived under house arrest, and 4 years later, when Mary died of a terrible virus, influenza in those days, Elizabeth became Queen. Her motto was Semper Idem, which means always constant, and that kept her true because she became not only the most successful Tudor monarch, but arguably the greatest British monarch of all time, because she defied convention, being female and being powerful. She also saved England from religious civil war and also from invasion, and at the same time, she managed to enable the arts to flourish. Shakespeare was one of the beneficiaries of her reign. So that is the background to where I started. She was so inspiring to me, as you can imagine. What an amazing woman she must have been. So back to me. So now let's move forward. Now I'm 6, 7 years old. My first school was junior school, the equivalent of an elementary school, St. Peter's Junior School in a little village in South Nottinghamshire, in the middle of England called Reinventing. Now, this is not quite as grand as the Tower of London, as you can see. There's a little junior school built in the 19th century where I spent a couple of years. But it was a nice education, a very quiet little village education. I then went to secondary school, sort of the equivalent of late elementary high school, in what was actually Rushcliffe Comprehensive School in those days. It was a non-private school funded by the government. Really, it was more like a school attached to a leisure centre, because its primary claim to fame at that point was it had this amazing leisure centre where we could all go and swim or do sports. Well, not me, because I couldn't swim, but play sports and all that kind of stuff. So as you can imagine, it was not a great background, but it was a friendly, lovely place, and I had a great time there for 7 years. Nottingham, of course, is famous for much more than Rushcliffe School, me, and St. Peter's Junior School. It's famous for Robin Hood and Maid Marian, and that's a statue of Robin Hood, which has been next to Nottingham Castle now for, over about half a century. Now, that's me not when I was a young girl. That was me a couple of years ago when I took a trip back to Nottingham after many years away. And I insisted on having my photograph taken next to Robin Hood because I remember being there when I was indeed a little girl of about 6 years old. The other thing I want to say is that about this time, I became interested in a kind of science, which is the science of stamps, or philately. And here's Sir Rowland Hill, who modernised the British Mail Service and was actually responsible for originating the first ever postage stamp in the world, certainly the first UK postage stamp, which in 1840s, the penny black. Now, when I first gave a part of this talk and Dr. Lewis was in the audience, he came up after me and said, well, yes, Mary, that's all very well, but what's your favourite stamp? And I remember that, Dr. Lewis. Well, here's the answer to that question, Dr. Lewis. My favourite stamps were actually made in a year of my birth, 1964, and they celebrated Shakespeare's birthday. At that point, in 1964, which is April the 23rd, 1964, which many of you will know was the 400th anniversary of Shakespeare's birthday. Now, what's so special about these stamps is that they were actually one of the first set of commemorative stamps whereby the events depicted were not about royal events or festivals of Britain or anything regal like that. They were actually about somebody else. And that's what made them so special. But of course, for me, they were special because they hark back to Elizabeth I and her reign and her patronage of the arts. At the same time, when I was in Nottingham growing up, I became friends with a relative of Sir Rowland Hill. Her name was Allison Hill. I can't tell you exactly how she was related, several generations apart, obviously, but she was definitely a relative. She was a colleague in science classes. She was an artist, a poet. She had terrible mood swings, and it was only actually several years later that I found out that she actually had a diagnosis of bipolar disorder, which she kept hidden in her teenage years. So what happened to me? So I left high school, I left Rushcliffe, the Leisure Centre, and I went to Cambridge University to do medicine. And there's a picture of King's College Chapel, which is the most famous probably picture of Cambridge. And there's a picture of me. You can tell it was the 1980s, can't you? Look at that hair. What did I think I looked like? But anyway, there you go. So that was me on my graduation from Downing College, Cambridge, in the 80s, in the middle 80s. And there's a picture of our graduation picture. This is just the 100 or so people in Downing College that year. And you can see the blue arrows pointing to the woman with the ridiculously long, frizzy hair. In those days, I was the only one, as everybody else was wearing those sort of, had those different short hairstyles. But there you go. Now, I think what you'll see about that picture, other than it being very grand, we're all wearing the black and white gowns, etc. You'll see that there are lots of people who are white, Caucasian. Very few. I think there was one person who was non-white in that whole year. And although there were several females, very, very small minority in comparison with the males. And I have to say, we were only the third year, just the third year in which women were allowed to attend that college. So this is the 1980s, and it was only 3 years that women are actually allowed to attend. As you can imagine, it was a very sexist, very racist atmosphere, very unpleasant in many ways. So how did I manage to get through? Well, I took solace in the sea slug. This is a Plisia, which is, many of you know, is a sea slug, and Eric Kandel and others made it famous because it became the first creature whereby the whole neural network and emergent behavior, connections, relationships were identified. I was fascinated by this, and I learned a lot about it in my third year doing zoology. I did a mixed degree of medicine and zoology, which was possible in those days. So that kind of gave me inspiration, it kept me focused, and it enabled me to cope with the sexism and the racism which was rife in those days. The other aspect of my life at that time which really helped me cope was this person, Jane Austen. Jane Austen, and I know I don't need to introduce Jane Austen to you, but I want to highlight why she's important to me. At that point, so I'm sort of in my early 20s, I read virtually all of Jane Austen's works, all her major novels. And like us all, we all love Pride and Prejudice. I like Sense and Sensibility. But my favourite book of all was Persuasion. And the reason I like this book, it was her last novel, the most mature novel, and it's about a very sad story about Anne Elliot, who comes from a wealthy family, but is persuaded to drop her engagement, cut her engagement with a then penniless sea captain. And what transpires is her family becomes poor and he becomes wealthy. Years later, they meet again, and eventually she's able to marry him. The point about Anne is that, as Jane Austen says, she's a very, very wonderful person, almost too good for Jane Austen, as Jane Austen notes there in her letter. Basically what Anne does, she's a true constant, just like Queen Elizabeth I, Semper Idem. She is a constant. Her behaviour remains the same. Even though everyone else around her changes, the times change, she remains the same. And eventually people are persuaded to really appreciate and love her. And that was another inspiration to me. So with that in mind, I was able to continue in my pursuits of science and enter the Maudsley Hospital, which, as many of you know, is one of the most famous, and still is, one of the most famous places in Europe for training in psychiatry. And so that's where I began my journey, really getting interested in emotion in the human brain, very much inspired by William James, as we all know, a very eminent scientist, physiologist, who postulated that emotions arise from physiological responses in the body and the brain. Now, at that point in time, of course, we didn't have neuroimaging. We certainly didn't have FMRI everywhere, as we do now. So to think about trying to image emotion in the live human brain was a bit of a giant leap. That's an endeavour which I tried to take. However, along the way, you may be asking, well, what happened to Allison, my friend? Well, it turned out that Allison was also interested in psychiatry too, and she qualified at the same time as me as a psychiatric nurse at University College London and also at the Maudsley Hospital. And we had a very good relationship. We rekindled our childhood relationship. In 1993, she and her boyfriend decided to go on a round-the-world year and a half long trip, which is very exciting. About a week before that, she and I had agreed to meet. Unfortunately, at the last minute, I was called in because I was in jeopardy. I was second on call in psychiatry as a psychiatry resident. So I had to say, sorry, Alison, I can't make our reunion. This is a letter Alison wrote to me at that time, saying, sorry, we couldn't meet. When we get back, her boyfriend Dominic and she, when we get back, we'll meet up. I thought, fine. Now, you might wonder why I've kept this letter. The reason I kept it is because it's actually the last time I had any proper contact with Allison. Because what happened is that she went away on this year and a half long trip. She got back. I didn't really have much contact at all with her. In fact, I didn't have any direct contact with her at all at that point. The last thing I heard is that she'd moved up to Nottingham, and then I hear in 1996 she died. She'd taken her own life. She couldn't take it anymore. She and her boyfriend had broken up, and it had just pushed her over the edge. I never ever got to see her again, and that's one of the things I shall always regret. But you have to carry on, don't you? You have to kind of find it within you to pursue your ambitions, your dreams, and just get on with life, no matter how sad and awful it is. Marie Cury, at that point, she was somebody we all know her story. I had been transfixed with her at one point in my earlier life when I was in secondary school at Rushcliffe, the leisure centre. I wrote a story about her. I wrote an essay about her. I think everybody knows that, you know, she was the first woman to win a Nobel Prize, but she was also the first person to win 2 Nobel Prizes as a woman. That's remarkable. And she's the only person to have won 2 Nobel Prizes in different science fields, still the only person. That's astonishing. And she was a woman at the same time. Also, the other person who began to inspire me at that time when I was in the Maudsley and the Institute of Psychiatry was Geoffrey Gray. Now, Geoffrey Gray, many of you may remember or may know him through his wonderful work on neural circuitry of approach and avoidance behavior, stemming from rodent work, but also pertaining to human work. He's one of the originators of the BISBAS, the behavior inhibition and activation scale. And he was a marvelous mentor to me at that point. Just when I needed it, he was amazing. The story actually is that in 1996, just after Allison had died, I got a phone call. I was in my office in the basement, this windowless little basement room, because that's where we female scientists were placed in the Maudsley. I was sharing an office, a basement, little office without any windows. I got this phone call saying, hello, it's Geoffrey Gray here. Would you be interested in taking part in a neuroimaging study with me? And I said, yes, please. Being really famous at the time. He went on to make some marvelous discoveries about human consciousness. And this is a book that was published posthumously by him. But he and I and many others on the team got together and really were able to do some studies, and where we tried to ask the question, how are different emotions embodied, encoded in the human brain. And this is my first ever neuroimaging study, which I was fortunate enough to get published in the journal Nature. And this is where we basically identified the seat of human disgust. The reason this was important, the reason it got attention, was because at that point in time, everyone thought that the amygdala was the seat of all emotion. But of course, we now know that's not true. But at the time, this was the first real study to show that. But then I thought, wow, this is all going rather well. And I got interested in applying that knowledge to trying to understand how do we actually try to unravel the mystery of the bipolar disorder brain and mind. What are the neural circuits that allow us to try to identify what goes wrong in people with bipolar disorder? Now at this point, and we're talking now in the early 2000s, Richard Lane, who is there with a white coat on, Richard Lane had just moved to Arizona. And Richard had come to visit London, had come to visit me and a couple of other people at the Institute of Psychiatry. He stayed with myself and Mike in our house in London. And over dinner one night, he said, I don't know, Mary, I don't know about you, but I would really find it helpful if there were kind of a roadmap, if there were a template for emotion regulation in the human brain and the neural circuits thereof. So we decided to write a series of review articles, and we enlisted support from Scott Rausch, who's there in the top right-hand corner now. Many of you now know he's the chair in McLean. And then Wayne Drevitz, who at that point had just left Pittsburgh and had moved to NIH, so a bit of a connection. But at this point, I was firmly seated in London, and so we wrote these reviews, and they were published in Biological Psychiatry, and they received a lot of attention and continued so to do for a few years after that. But perhaps the most important thing about 2003 to me was ACMP, when I met this gentleman, Dr. David Kupfer. And basically in ACMP, in a few months after that, he made me an offer I couldn't refuse. And to cut a long story short, over the next year, with the support of many kind people, Laurie Batkai, as was then, and many other people in the department, I and Mike, we came to America, and I became full-time faculty in July 2005. And I think without any doubt, these last 15 years have been the most productive and happiest of my professional life. And I have David Kupfer and many other people to thank for that. And now David Lewis as well, and who's a wonderful mentor to me. So I was on a roll, right? I'd done these nice reviews. I'd come to America. I could finally speak the language. I was now trying to publish more work. And Cecile Ladisser, whom actually had contacted me a couple of years before I came to Pittsburgh at a conference, Biological Psychiatry in actual fact, she and I worked together at that point, and we wrote, along with Wayne Drevitz, another review which has become influential to some extent. And this was a review where we tried to understand and unravel in more detail the intricacies of emotion regulation, identifying two kind of main areas of emotion regulation, the kind of automatic feed-forward implicit, and then the more effortful top-down explicit. And this was going very well, and I was then starting to apply this to trying to understand bipolar disorder, as I had already set out to do. And at this point, Emilio Versace and George Almeida came to join me in Pittsburgh and work in the lab. And we together published a series of papers. This is just one example I'm showing you here. We published a series of papers where we were showing what was specifically abnormal in the prefrontal cortical amygdala relationships, coupling, regulatory systems that were specific to bipolar disorder and not shared with other disorders such as unipolar depression. And here's just one example, as I said. This is a functional imaging paper where we're showing that bipolar disorder, unlike unipolar disorder, unipolar depression, bipolar disorder is characterized by this lack of regulation, if you will, lack of coupling, almost like an absence of coupling between prefrontal cortex and amygdala. So this, and as I say, there were many other papers as well. Amelia is an expert in diffusion imaging, white matter pathology, and she has produced and published some beautiful studies showing similar things, but focusing more on the white matter. So this was all very well, but there was a huge problem, wasn't there, really, because this focus on dysregulation, affective lability, great, we're showing specific abnormalities with bipolar disorder, specific abnormalities in amygdala prefrontal cortical circuitry, but every disorder every mood and anxiety disorder is in some way characterized by affective lability. Even risk for mood disorders and anxiety disorders is characterized by the same thing. And what is more, everyone was starting to get sick and tired of the amygdala prefrontal cortical yin-yang story. It was kind of getting old. So what do we do? I mean, where do you begin? Where do you next move if you're trying to understand and really nail down what's so special about bipolar disorder? Well, here I want to now switch tack and just talk very briefly about somebody else who has inspired me over the years. And this is Fanny Luhama, whose actual catchphrase was, "I'm sick and tired of being sick and tired." For those of you who don't know her, Fanny was a civil rights leader in the 60s in this country. She co-founded the Mississippi Freedom Democratic Party because she was sick and tired of the Democratic Party at that time not being friendly enough, not being inclusive enough with regard to people who weren't white. She persisted in her endeavors as a politician despite actually being tortured and beaten in jail because she was black. Basically what happened, she was coming back from actually having registered to vote, finally managed to register to vote. She and her female colleagues sat in a bus shelter which was for whites only. They were arrested by the police and she was beaten up basically by other inmates at the police's orders in jail, so badly that she lost an eye, she had kidney problems and she couldn't walk properly after that. But she persisted. She persisted, and so much did she persist that she was actually feared by President Lyndon Johnson, who refused to actually be on the same platform with her. And while his speech in one convention was being broadcast, she stole the limelight. And the television cameras went to her and recorded her. She got as much attention as he did. She persisted and went on, reinvented herself, and went on to do other things before she died, sadly, of cancer in 1977. Talking about changing tacks, reinventing, that's what we have to do in bipolar disorder to try and understand really where the functional abnormalities were. And I really just want to highlight the sort of strategy that we've been using. Bipolar disorder is characterized not just by affective lability, but by high sensitivity to expectation of reward, by high impulsivity, sensation-seeking, thrill-seeking. All of these are evident plus in people with bipolar disorder. But they're also, we know that these behavioral traits also predispose to hypomania in people who haven't yet developed bipolar disorder. So there's something rather special about this constellation of different response tendencies, if you will, behavioral traits that seem to be specific to predisposition to bipolar disorder and hypomania. And around about this time, I was fortunate enough to be able to work with Erica Forbes in the department, and Henry Chase, who joined me as a postdoc in those days in the lab too. Now, what Erica had developed was a really lovely reward processing task, which was really special because this task, unlike many others in the field at the time, was easy to perform by children and adults alike. And it also captured this reward expectancy, so when you're anticipating reward, as opposed to when you're actually receiving reward or not. And as I've just said, bipolar disorder and people who are at risk of becoming bipolar are characterized by this high reward sensitivity and reward-driven impulsivity, which basically are associated with high expectation of reward. Furthermore, we know that in reward gambling, when people are goal striving, people with bipolar disorder and people who have high levels of these behavior traits, response tendencies, are more likely to become hypomanic in various degrees. So when you put people with bipolar disorder, proneness to bipolar disorder in goal striving, reward expectancy conditions, they are more likely than in other conditions to have a hypomanic episode or have their hypomania symptoms become worse and develop hypomania-related impulsive behaviors, reward-driven impulsive behaviors. So the task really, as I say, has its really important anticipation condition. And what Henry and I were able to do was to develop this task a little bit to kind of broaden out this anticipation expectancy phase. And I'm not going to give you all the details, just to suffice to say that in this task, there are lots of different conditions where you're expecting something and you can either win or nothing will happen. You can either lose and nothing will happen, or you can win or lose, or just nothing's going to happen. So that enables us to basically look at a whole range of different reward expectancy contexts, ranging from, gosh, I could really win something, to gosh, I could only lose, or either way, or nothing. We've published a lot of papers in bipolar disorder on this task, and Erica's previous version of the task. But I just want to highlight one paper where we were able to show that people who haven't yet got bipolar disorder but are vulnerable to bipolar disorder, to getting the disorder, because they have high levels of this impulsivity, sensation-seeking, reward sensitivity. Those people, along with the people with bipolar disorder who we showed in previous studies, what they do is they have higher levels of activity in this area in the brain during, as the expected value of reward increases during the reward anticipation. So the more likely you are to get a reward, the more this activity increases in people who are bipolar disorder prone. And this area is the left ventrolateral prefrontal cortex, or left VLPFC for short. We have replicated this finding in an independent sample of people at risk for bipolar disorder. And this is work with Kale Edmiston and Jay Fournier. Kale is a junior faculty on a K Award now in the lab, and Jay is on the route to becoming associate professor. And Kale and I, on a completely independent sample, were able to replicate this finding. What is more, we were able to show that in the combined sample, that having this pattern of increased left VLPFC activity during expectation of reward, associated with this impulsivity flavor, this impulsivity response tendency, actually is related to your risk of becoming hypomanic or getting a bipolar disorder later on. And this is as measured by the mood scale, which many of you know is a lifetime vulnerability to bipolar disorder scale, developed by Ellen Frank, David Kupfer, and colleagues. So here we are, showing that not only does this signature is there in people at risk, but it actually relates to future risk of getting bipolar disorder if you haven't already developed the disease. And that, of course, brings me to talk very briefly about what does the left VLPFC do. To cut a very long story short, the take-home message is that the left VLPFC is key area involved in decision-making to attend to have preference for, and to optimize your chances of getting immediate future rewards. So in any different context, the left VLPFC kind of biases you towards wanting to go for immediate, soon, early rewards, no matter how big or small. And so this is a kind of final link in the chain. I told you, that the amygdala and prefrontal cortex is involved in affective lability, emotional dysregulation, but there's this key other strand linking the reward circuitry, the left VLPFC, that predisposes much more specifically to hypomania and ultimately to bipolar disorder. So they're both important, but the left VLPFC, etc., is much more specific to hypomania, which of course, as we all know, is the pathognomonic feature of bipolar disorder. And that has got us all towards thinking about treatment targets, which is one of the key things I've always wanted to do, and as Dr. Lewis alluded to in the introduction. So here, we're thinking we're really on a roll now, and there's this new neuromodulation, non-invasive neuromodulation technique, which was new at the time, transcranial direct current stimulation, which is easy. You can even buy the kit off Amazon. It's that easy. You just stick 2 electrodes to your head, and then you can stimulate or down or activate a region. And so we use this technique to try and perturb the circuitry in people with hypomania and bipolar disorder. And here is essentially what we're trying to show. We're trying to show that people with bipolar disorder, or who are prone to bipolar disorder, they have this heightened activity in the left ventrolateral prefrontal cortex and reward circuitry, and this predisposes to hypomania. If we can damp that down with inhibitory stimulation, we should be able to reduce risk symptoms of hypomania, right? Well, we did show this, and this is work, as I've just indicated, with Michelle Bertocci and Henry Chase in the lab. Here, what I'm showing you is across a large group of people, with healthy people and bipolar disorder, when we try and inhibit this region in the brain with inhibitory stimulation on the x-axis, after the event, we are reducing the intensity of their hypomania negative affects, so their irritability, anxiety, arousal. But there's another problem. TDCS is highly controversial. Many would argue that it doesn't work at all, that it's just epiphenomena, it doesn't get focal in the brain, etc., etc. You name it, the criticism just kept on coming. So yet again, I'm here at a crossroads. What do I do? Do we continue? Do we persevere with this controversial technique? In fact, we wrote a review about this here. This is the third one. Or do we try and focus on a technique that is better, that is more focal? And this, I just want to really highlight the inspiration here, RBG, who I don't need to introduce her. She is well known throughout the world. She was an inspiration to us all, obviously, and to me at this point in time, totally, because despite all the odds, despite her illness, she persisted, she persuaded others, and she continued to do what she wanted to do. And she sadly died, as we all know, far too soon. So with Fabio Ferrelli in the department, he and I have persisted, and we have now focused on using Peterborough Stimulation, which is much more focal than TDCS, and have used this to basically answer the same question, whereby, can we tackle this circuitry and can we reduce predisposition to mania? And what I'm showing you here are 3 figures from a grant application, as I should say, where we're showing that here's the model. We're going to give people with bipolar disorder and healthy people this stimulation, inhibitory stimulation. We're showing that it's focal. If we target the left VLPFC as opposed to a control region, and we're showing here that indeed, if we target this circuitry, downstream effects in the reward circuitry are much more pronounced in people with bipolar disorder and healthy controls. Now, I say that these are figures from a grant application because Fabio and I have persisted for about 2 years to try and get funded for this research. And I am pleased to say that now we are told that they're preparing the NGA, I hope. It's on the list for funding. We got a very fundable score. Wait for the NGA is basically what our program officer has told us. So persisting pays off. Finally, I just want to highlight other inspirational people in the department, and I just want to tell you about the Neurochicks, who many of you will know. The Neurochicks are a Pittsburgh institution now. Here we are, we have Atika Hall, Bea Luna, Erica Forbes, Anne Germain, Colleen McClung, and Cecile and myself, Cecile Ladisser and myself. And we are a group of female, more senior neuroscientists who meet whenever we can, which is not as often as we'd like, to get together, self-support, etc., and to carry on motivating each other. And it's been very important, especially during the shutdown, I have to say. We've done these virtual meetings. And many of us are interested, we have a lifespan perspective to neuroscience, to effective neuroscience, and we're interested in the development, early life development of predisposition to these disorders. So that is a segue to tell me to basically, finally, in my last couple of slides, I just want to highlight 2 other inspirational women in the department with whom I have the pleasure to collaborate. And this is Allison Hitwell and Ayla Bani-Hashemi. Allison and I have been funded now, we've won our 2nd award from NIMH, to examine neural behavior relationships in the infant brain. So we're talking 3-month-old infants, and how the caregiving environment can actually shape those critical early relationships. So this is the kind of structure. So we're having, the caregiver at the left here, how that shapes the development of the baby's brain and how that in turn impacts behavior. And on my last slide, I just really want to show you some sort of most up-to-date findings. We published a couple of papers, but the most up-to-date findings are these. We're showing that despite the fact that we all know we replicated a relationship between high caregiver depression and low infant smiling, we're also showing that a specific pattern of baby brain connectivity protects against that deleterious relationship. And this is where the baby's brain here, the salience network, which is involved in self-emotional salient processing, if that is less connected and the regulatory prefrontal cortical situated executive network is more connected, the combination of that actually protects against this otherwise negative relationship, deleterious relationship, and the baby is less likely to be a low smiler. So we're getting a signature of resilience, if you will, in the infant brain. And at that point, I just want to say that we've replicated that in 2 samples. We've shown it in 1 sample and we've replicated it in another sample. And at that point, I just really want to take the opportunity to thank everyone in the lab. This is my lab at the moment. I want to thank all the wonderful staff members at the top there and their pets. I hope you can see some of their pets. And then at the bottom, the marvelous faculty, postdocs, and students in the lab. I also really want to highlight all my wonderful collaborators and the funding streams from NIMH, NARSAD, and the Pittsburgh Foundation. And finally, I want to thank you for your attention and my muses here, all four of them, who accompany me every day at home. This is my home office, and they're always there looking out for me. But thank you very much for your attention.
Mary, thank you very much for an outstanding presentation. We can have questions from the group here, and Mary will ask you to repeat the questions. And Jeannie, do we have another slide in here that tells people how to text their questions in that we can put up?
Yes.
So that.
Yes, we can.
Okay.
I'm also putting it in the chat room.
Are you?
Yeah, I don't see any in here yet. Oh, here we go.
Yeah.
Okay, Mary, first question. Do your findings help us understand the cyclical nature of bipolar disorder 1?
The question is, do the findings help us to understand the cyclical nature of bipolar disorder 1? Well, my answer would be yes, they do, and this is the reason. Bipolar disorder can be considered, I mean, it is a cyclical disorder. Hypomania is pathognomonic. There are several computational models now which are out there showing that hypomania might come at some point in the disorder. But what happens when the reward expectancy, et cetera, gets thwarted? What happens is that people get pushed into depression. And so the cycle, and then depression finally wanes, treatment or otherwise, and then hypomania starts again. But the key thing is, if we don't understand hypomania, we're never going to understand the beginning of that cycle. So by understanding or trying to understand the neural basis of hypomania, we're at the start of that cycle, or somewhere early on in that cycle. Then we can start to understand, okay, what happens when things get thwarted and then getting depression. So which part of the brain kicks in to stop the hypomania vulnerability, sort of almost damp down to push towards depression. So, you know, the question I hope is answered in the sense that we have to understand where the cycle starts if we're going to understand the cycle. And the cycle, by definition, starts with a hypomania, because until then, people don't have a diagnosis of bipolar disorder.
I have a question for you, given your new grant coming in. Congratulations.
Thank you.
Can you tell us a little bit more about how TDS, in terms of where you can direct this modulation to, differs from TMS? Can you get it deeper structures? Can you get it to the midline? So,
yeah, so TDS is...
can you repeat the question?
Yeah, yes, sorry, the question is, what's the difference really between TBS and TMS and TDCS? Why is TBS supposedly better? I think that captures the question. So just to go back a stage, transcranial direct current stimulation, very popular, very easy, painless. But the problem is it's thought not to really have a very much of a focal effect. It's too diverse, too dispersed throughout the cortex. There are ways of getting around that with different montages. The thing about TBS, it's if you will, it's the more recent version of transcranial magnetic stimulation, TMS, and it's much briefer. It's focal like TMS, but it is much briefer in the way it's administered. So it's as effective as TMS, which is much more effective than TDCS. But TBS is much briefer in its administration, and therefore it's tolerated better. Because TMS is much more focal, it's also more painful. And therefore, the problem with TMS is that it might not be tolerated because it's painful. It might work, but it's not tolerated. TDCS, tolerated, but might not work. TBS works and tolerated better. And the physics of that is that it's just a faster waveform. It's theta burst, and it effectively gets to the deep structures in the same way as TMS does.
You could remind the audience that they can text in questions
yes.
to that. I have another question while they're doing that. So, you know, at the outset, you beautifully set up the idea of higher reward sensitivity and reward-driven impulsivity as key element in some. Could you speak a little bit to the extent to which you think those are
transdiagnostically important issues, and they come up in addictive disorders, for example.
Yeah. So the question is reward sensitivity, impulsivity, sensation seeking. These are likely probably transdiagnostic. So to what extent are they evident in other non-bipolar disorders? And yes, absolutely right. And I'd be the first to admit that. And I think what I like about this aspect of the research now is that it really is its transdiagnostic approach. The fact that there are many disorders that share similar features to bipolar disorder, but have different endpoints. But at the beginning, they may have a very similar neuropathophysiology. Addictions, substance use disorders are a very good example, and in actual fact are highly comorbid with bipolar disorders. So it makes sense that there's going to be some shared pathophysiological mechanisms. But this also, I think, gets to the point about why is understanding psychiatry in terms of transdiagnostic approaches really important? Well, and this might be controversial, but I think that it's the way to go because our whole classification system really needs rebooting. And we're basing it on old-fashioned, if you excuse my expression here, traditional views, which are behaviorally based. And I think it's classical, thank you, classical, which are classically based. It's time now to try and understand psychiatric disease in terms of underlying pathophysiology. I mean, it's an obvious thing to say, but it's hard to do. But having the transdiagnostic approach is one way to do it, because we can look at groupings, we can look at different dimensions, where the beginning points are, where the end points are, and say, well, these two dimensions look the same, these two don't. The endpoints might not actually be bipolar disorder and substance use disorder. It might be, reward sensitivity plus substance use, or reward sensitivity without substance use. There are going to be different ways of classifying these disorders based on understanding of the underlying pathophysiology. So, to me, it's critical to try and understand, again, it gets back to where do you start and where do you end. Starting is with dimensions, endpoints develop as a result of the development of those dimensions.
I have one additional question, and this one will be challenging.
Okay.
That's why we asked you to talk. So you've done a great job of telling us about, from the past, inspiration and the importance of perseverance and persistence. When you look into the future, and for individuals who have a similar motivation, you have to understand the nature of an illness in order to try better diagnostics and therapeutics. What do you think, where's the future for actually being able to most robustly and with required level of resolution actually see psychopathology? So is FMRI going to continue to have legs in the future to really give us targets for therapeutic interventions, or do we need new tools, and if so, what do you think those are? And you can say anything you want now.
Thank you.
It's a pretty broad question.
Well, thank you for that really easy question to answer, Dr. Lewis. And so the question is, what's the future of psychiatry? And understanding biological mechanisms, is FMRI or everything? Well, how can I answer that question? I think the future, this is my personal opinion, obviously. My answer would be, we have to understand the brain basis or the pathophysiologic mechanisms if we're ever going to be able to develop new treatments that actually target the very organ that psychiatry is about, which is the brain. Now, I think in so doing, we need more than FMRI, obviously. We need much more than human neuroimaging. And this is why I would just like to give a big shout out to my other collaborators who were on my acknowledgement slide, but I didn't sort of mention by name. These are the people who work in non-human primate research and who are doing wonderful work, so Tobias Teichert and others, Carl Allison and others. And essentially, we need to come together and think in creative ways of combining different techniques, different human imaging techniques as well, as animal models, to really dive deep and try to understand the pure direct measures, the direct measures of the actual tissue itself. Dr. Lewis, I would argue, I think you would agree, trying to get to the the actual physical basis by having direct measurements of activity in models which allow that, and then linking that to human imaging techniques, which at best are only ever going to be indirect measures of neural function at the moment. And then sort of having that perspective to really truly understand the in-depth mechanisms. And only then can we really start to identify those targets for treatments. Alongside the wonderful clinical measures that we all do. I'm a psychiatrist. I was brought up to do psychiatric evaluations based purely on patient history and collaborative history. No neuroscience involved necessary. I think no one is going to say that clinical evaluations are not important. Of course they are. They're essential. But it goes hand in hand with the biological perspective.
You actually answered a question that just came in, which was what's next for you. But there's one additional question that I think will be a great way to end this session. The question is to integrate the questions so far and Allison Hill's tragic death. How can reward disruptions help us understand the high rates of suicide and bipolar disorder?
So the question is, linking back to obviously stuff in my facts in my history, how can understanding the neural basis of reward sensitivity help us understand the suicide rate in bipolar disorder? Well, I think this links back to your one of the earlier questions, Dr. Lewis, which was how can we understand the cyclical nature of bipolar disorder by understanding reward sensitivity and hypomania. Depression is the context in which most of the suicides, not all, but most of the suicides in bipolar disorder will obviously occur, and certainly did for my poor friend Allison. So I think, going back to the beginning is the beginning of the cycle, beginning of the will. We need to get in there at the beginning, early in development, ideally, look at that predisposition and know that downstream, if you have this high reward sensitivity, neural mechanisms thereof, that is going to push you into the next stage of the will, the cycle, which could be the depressive phase, which is a natural physiological defense mechanism against the hypomania. There comes a point when the brain gets so exhausted, it can't cope with this hyper-aroused, hypomanic state. There is a natural mechanism. I've seen it, actually. A story, very brief. When I was a resident, actually before I was a resident, when I was a medical student in Cambridge, I saw someone who was absolutely, frankly manic for a week, but that's a bit longer. And the next time I saw him, he just naturally become depressed. This wasn't because he'd had ACT or any sort of drastic change in medication. He just naturally, his brain couldn't cope with it, become depressed and then remained depressed for some time. Really psychomotor, totally, depressed, the full gamut, as it were. So, by understanding the reward sensitivity, the brain predispositions to that, it's the beginning of the cycle. And you can say these people are going to become very depressed later on if this isn't tackled early. Reward for hypomanic today, but depressed tomorrow. And then if they're depressed, and very depressed, left untreated, that can precipitate suicide. So I think that is one of the reasons why there's such a high suicide rate in bipolar disorder, because it's hard to detect because of the hypomania masking. It comes later down the cycle. People don't always connect the two. There hasn't been until recently an obvious link between the two. People don't necessarily assume that suicide is going to happen in people with hypomania. So as long as they're a little bit okay, it's going to be fine. And that's undoubtedly what helped Allison continue as long as she did. She didn't probably get the treatment she needed because she had a kind of sunny disposition, which was really the hypomania. So that's how I would answer that question.
Thank you very, very much.
Thank you.